Animal cell formation is a fascinating process that showcases the complexity and beauty of life at a microscopic level. Let's dive into the world of cells, specifically focusing on how animal cells are formed and what makes them unique. At the core of animal cell formation is the process of cell division, primarily through mitosis. This is how a single cell divides to produce two identical daughter cells. It all begins with the parent cell, which contains a complete set of chromosomes. These chromosomes are made up of DNA, the blueprint of life, which carries the genetic information necessary for the cell's functions. Before mitosis occurs, the cell goes through interphase, where it prepares for division. During this phase, the cell grows, duplicates its DNA, and ensures that all the necessary organelles are ready for the new cells. Think of interphase as the cell's way of getting ready for a big event. Everything needs to be in order before the main show begins. Once interphase is complete, the cell enters mitosis, which is divided into several stages: prophase, metaphase, anaphase, and telophase. In prophase, the chromatin condenses into visible chromosomes, and the nuclear envelope begins to break down. This is when the cell starts to look like it's gearing up for action. Next comes metaphase, where the chromosomes line up at the cell's equator, attached to spindle fibers that help pull them apart. This alignment is crucial because it ensures that each new cell will receive an identical set of chromosomes. It's like lining up for a race, making sure everyone is in the right position before the starting gun goes off. Then we move to anaphase, where the sister chromatids are pulled apart and moved to opposite ends of the cell. This is where the magic happens. Each half is now a complete set of chromosomes, ready to be packed into the new cells. Finally, in telophase, the chromosomes begin to decondense back into chromatin, and the nuclear envelope reforms around each set of chromosomes, creating two distinct nuclei. But we're not done yet. After mitosis, the cell undergoes cytokinesis, which is the final step in cell division. This is when the cytoplasm divides and the cell membrane pinches inwards, ultimately splitting the cell into two separate entities. In animal cells, this is often achieved through a structure called the contractile ring, which helps to ensure that the division is clean and efficient. What's truly remarkable is that this entire process is tightly regulated by a series of checkpoints and proteins that ensure everything goes smoothly. If something goes wrong, it can lead to issues like cancer, where cells divide uncontrollably. This highlights the importance of understanding cell formation, not just for biology, but for medicine and health as well. Animal cells are also unique in their structure. Unlike plant cells, they lack a rigid cell wall, which allows for greater flexibility and a variety of shapes. They contain various organelles, each with specific functions. For instance, mitochondria are the powerhouse of the cell, generating energy, while ribosomes are responsible for protein synthesis. The endoplasmic reticulum and Golgi apparatus work together to process and transport proteins and lipids, ensuring that the cell functions properly. In summary, the formation of animal cells is a complex yet beautifully orchestrated process that involves careful preparation, precise division, and intricate regulation. Understanding this process not only sheds light on the fundamental building blocks of life, but also opens doors to advancements in health and medicine. The next time you think about cells, remember the incredible journey they undergo to form the very essence of living organisms.